factors in some cells, but not in other cells. Those transcription factors might turn on genes for specification in one cell, but not in any other cell in the embryo. And again, we find this in the tunicate embryo. So here we have a cell fate map of the tunicate embryo. And what we see in this fate map is that there is a cell in the vegetal posterior, the B41 cell, actually two cells, since both sides are symmetrical. The B41 cells make all the cells in the tail of the embryo. They are autonomously specified to become tail. And if one looks at the embryo, one sees that the precursors of the tail, starting with that B4 cell, these cells all have a different colored cytoplasm. They have what's called a yellow crescent cytoplasm. Now, this cytoplasm will be found in all the tail muscle precursor cells. Now, if you take a tunicate embryo at this 8-cell stage, and with a glass needle, you separate the cells into those 4 groups of 2 cells each, what you find is that the B4 cell will make muscles and endoderm. Other cells will make other cell types. The brain, however, the central nervous system, is made by induction, so it is not found when you separate those 4 groups of cells. But B4.1 cell, that becomes muscles, and only that cell gives rise to muscles. And if you take that cell away from all the other cells of the embryo, and you stain for a muscle-specific protein, such as acetylcholinesterase, which is shown here in black, the only cells that have that are the cells derived from the B41 blastomere. Also, if one was to delay division, again with a glass rod, and push some of that B41 cytoplasm up into the cells which normally become ectoderm, those cells can also make muscles. It was known for decades that there was something in that cytoplasm, in the capital B4.1 blastomere, that caused muscle formation. What was it in the cytoplasm? Well, one group of researchers found an mRNA that was only found in those cells that were destined to give rise to the muscle cells. It was originally found in the vegetal pole of the embryo, just like that yellow cytoplasm. During fertilization, it migrated up into the B4.1 blastomere, and it was found in those cells. This mRNA was found in those cells that were destined to become muscle cells. So what did they do? They did the technique of find it, lose it, move it. So here we see the finding it. They found that this was an mRNA that was found in those regions of the embryo that produced cells destined to become muscle. So they found it. They called this mRNA, by the way, macho mRNA, macho 1, because it made muscles. So it was found exactly in the right places in the cytoplasm for muscle formation. Then they got rid of macho 1 mRNA. They degraded it with an antisense mRNA, and when they degraded it, no muscle cells were made. Then they moved it. They expressed macho 1 mRNA in cells that would normally become ectoderm, and these cells became muscle-forming cells. So find it, lose it, move it. They were able to show that macho 1 mRNA made a protein, a transcription factor, which told the genes to make the muscle-specific protein. This is autonomous specification. Only that one set of blastomeres, the capital B4.1 blastomeres, had macho 1 RNA. Those were the cells that became muscles. So that's autonomous, sometimes called mosaic specification. Now, conditional specification is done not only by transcription factors, it also involves paracrine factors. You have induction. One cell is telling its neighboring cells what to do. And as you've learned in other lectures, that paracrine factors work by binding to a receptor on their neighboring cell. When they bind to this receptor on the outside of the cell, it changes the conformation of that protein on the inside of the cell, giving that protein a new set of properties. Often it's a kinase property, which allows it to phosphorylate a protein, which eventually will phosphorylate or in some ways modify a transcription factor, which will then enter the nucleus and turn on or repress a group of genes. So here is how one group of cells can tell another group of cells what to do. It makes a paracrine factor. That paracrine factor is received by a receptor on the surface of another cell. It changes the internal conformation of that receptor protein, allowing it to have a new activity. That new activity will eventually modify a transcription factor, which alters the genes being expressed, alters the types of genes being expressed in that cell. We see this combination of transcription factor and paracrine factor going on in the sea urchin embryo. So here we have a sea urchin embryo over here. This is the first 4 divisions of the sea urchin embryo, ending up with a blastula. And what you could see is there's a particular division, to the 3rd division, which makes the micromeres. It makes these small cells. The division is such that about 95% of the cytoplasm remains in one cell, and 5% goes into these micromeres. We see some pictures of that over here, looking at nuclei and the cytoskeleton. There's a small cell that's being budded off. This cell, the micromere, is autonomously specified to become the skeletal forming cells of the sea urchin embryo. These micromeres are going to become the skeleton. They also have another property. They induce the cells next to them, such as the big cell that got 95% of the cytoplasm, to become the endoderm cells, to become the gut cells. So we have the micromeres being responsible for gut induction in a conditional sense, yet they are themselves specified in an autonomous way. They are specified by a particular transcription factor called beta-catenin, and here's the find it picture over here. Beta-catenin is found in high abundance in the cells that are those micromeres. It is found in high abundance in the micromere cells, slightly less abundance in the cells adjacent to them. If they get rid of beta-catenin, if they get rid of that protein, what you find is that hardly any of the cells become skeleton. Instead, they all become skin. They all become the ectoderm of the embryo. Nothing forms inside the embryo. And if they cause beta-catenin to be stabilized in all the cells in the embryo, what they get is basically a gut with skeleton. They get no ectoderm at all. So micromeres are specified to become skeleton by having this beta-catenin protein. What gives it the beta-catenin protein? There's a protein which is already in the vegetal cortex of the embryo, and it's called disheveled. Disheveled is a protein which stabilizes beta-catenin. So that beta-catenin is everywhere, but it's only stabilized in the bottom of the embryo, in the vegetal portion of the embryo. All the other cells, it's degraded. It remains on in the vegetal cells, in the micromeres. Those micromeres are important for inducing the cells around them to become gut. So here's what happens when you add these micromeres from the bottom of one embryo to the top of another embryo. What you get are 2 guts forming. You get invagination. You get the beginning of gastrulation at both sites. So in sea urchins, you have autonomous specification of the micromeres by the production of beta-catenin transcription factor and its stabilization in the micromeres that are destined to become skeleton. And it also causes the conditional specification of the endoderm, whereby these cells which can become endoderm or ectoderm are told to become endoderm. They're told to become gut cells by the micromeres. So that's conditional specification and autonomous specification. The third type of specification is syncytial specification, and this is what's found in fruit flies. The sperm enters an egg that already has structure. And when it fuses with the female pronucleus, when the sperm nucleus and female pronucleus come together, they will divide without cells forming, so that the entire embryo is full of nuclei, and cells will form around these nuclei only later. And this was specification which was studied by Jannie Nusslein-Volhard and Eric Wieschaus, who got the Nobel Prize for this, along with Ed Lewis, who looked at the later stages of differentiation and determination in fruit flies. So here is the nucleus expanding within the fruit fly without cellularization. Eventually, the nuclei go to the periphery and cells will form around them. Now, how does this happen? How do you get specification such that the cells of the head become different from the cells of the tail? In fruit flies, there are ovary cells called nurse cells. These cells make messenger RNAs, and these messenger RNAs travel through channels and they go into the developing oocyte. There are two of these messenger RNAs that get localized by their 3' untranslated regions. Bicoid RNA, the messenger RNA for the bicoid protein, gets tethered to the anterior cytoskeleton, while the mRNA for the nanos protein, nanos mRNA, the messenger RNA for the nanos protein, gets tethered to the cytoskeleton in the posterior end of the embryo. So bicoid is anterior, nanos is posterior. There are two other important mRNAs here. One is hunchback mRNA and the other is chordal mRNA, and those are found throughout the egg. Here's the bicoid mRNA tethered to the anterior cytoskeleton. That's where it remains. But it makes bicoid protein at fertilization, and the bicoid protein can diffuse through the embryo, but as it does, it forms a gradient, which is highest at the future anterior end and lowest at the posterior. And you could see the gradient of bicoid protein here, and bicoid mutants lack that gradient. Bicoid mutants don't make bicoid protein. Bicoid mutants don't have heads. The embryos are headless. So again, we have kind of a find it, lose it, move it situation. Bicoid mRNA is needed for head formation, and it's found in the right spots. Not only that, you could show that bicoid mRNA is the head-forming determinant by transferring it to different embryos. So here we have a wild-type embryo with bicoid mRNA in the future anterior. And there's a polarity. You have the acron, which is the most anterior portion of the head. You have the head, the thorax, the abdomen, and then the telson, which is the tail. So you have these regions of the embryo, starting with the most bicoid, acron, head, thorax, abdomen, telson. In the bicoid deficient embryo, where the mother is mutant for the bicoid protein and makes no functional bicoid mRNA to put in the egg, there's no head forming. There's no thorax. It's abdomen and telson. The telson, which is a terminal structure, gets put on both ends of this embryo, but it is a headless embryo. Lacks head, lacks thorax. Now, if one to take that bicoid deficient embryo and put bicoid mRNA into the anterior, you get a normal looking embryo. Acron, head, thorax, abdomen, telson. If you were to put bicoid messenger RNA into a bicoid deficient embryo in the middle of the embryo, that middle part becomes the head, and then around it is the thorax and abdomen. If one were to take a wild-type embryo, which has bicoid in the anterior, and put bicoid in the posterior, you get an embryo basically with two heads. Doesn't survive, but you get an embryo at least that starts forming with two heads. How is this happening? It turns out that bicoid and nanos can function as regulators of translation, and bicoid can also act as a transcription factor. So bicoid not only says make head, it inhibits caudal mRNA. It inhibits the translation of caudal mRNA, which says make tail. The Nanos protein, when it's made, can combine with other proteins, and when Nanos is there, it will block the translation of hunchback mRNA. And hunchback protein says,